Okay, welcome back to Bio 104, and we're going to start our first of the 4 themes or modules. They're called modules in Canvas just because that's how Canvas is structured. But to me, when I sat down to think about this class and what I knew I wanted people to learn, regardless of whether or not you are a bio major, I thought about themes that I could fit a lot of information into, and themes that are relevant to people's lives. First theme is cancer, and certainly in terms of relevance, it's difficult to find a more relevant topic in biology, besides maybe COVID these days, but it's estimated, I'm looking at figures here from the American Cancer Society, that 1,800,000 people in the United States will be diagnosed with cancer. Right? These are new diagnoses out of 300 million people, and that about 900 million or 900,000 men and women will be diagnosed, and a total of about 600,000 people will die from the disease. I myself had cancer 2002. I was diagnosed with testicular cancer, which is a pretty actually common disease among young men. I think 1 in 300 men will come down with testicular cancer. It's a pretty easy cancer to have. I had a pretty tough summer, had surgery and then 4 rounds of chemo, and then I've been cured for, it's been 18 years now. So yeah, cancer will affect, I think, 1 in 2 or 1 in 3 people. So you can kind of look left, look right. One of the 3 of you people will have cancer in their lifetime. So I wanted to teach you guys what you need to know to be able to understand the disease, what causes it, what doesn't cause it, how it's treated, why it kills you, and in order to be able to be a good caregiver, be able to be a good patient, maybe a good nurse, maybe, who knows. But needless to say, there are some underlying biological concepts that are really important in order to understand, in order to be able to understand cancer. Okay, and so that's where we're going to start. The theme, really, the cancer theme, it's about cancer, but really it's about cells. As I said in the last lecture, that all living things are made of cells, and so to understand cancer, you have to understand cells, how they reproduce, and what the different parts are. So that's where we're going to start. Theme 1. Now, keep in mind, I really recommend that you go to Canvas for the cancer theme or cancer module, and you look for the study questions. Download those study questions. What I recommend is that you go through, you read the parts of the book that are outlined in the study questions. You try to answer those questions for yourself. Come to lecture, or you listen to the lecture here online, and you try to match the 2 up and review the answers to those questions. Those are the type of things that are going to be on a test, right? And if you'll see as I go through this lecture that those questions are explicitly outlined on the slides. Okay. So here we see some bacterial cells. These are colored cells taken through a electron microscope. Right? Tiny little cells, but even bacteria are made of cells. Okay, so we see the bacterial cell here. If you look at the left, the axis shows size, and this is looking at micrometer, 100 micrometer size, which is the biggest, and then on the left side bottom, 0.1 nanometers. So a micrometer is a millionth of a meter, right? And nanometer is even smaller than that. So you're getting down at the subatomic level in the nanometer scale. Right. Again, we talked again a little bit about the idea that biological systems are organized. So atoms organize into molecules, molecules organize into macromolecules such as lipids, proteins, carbohydrates. Those are the things that cells are made out of, which then organize into different organelles like ribosomes. Viruses, very, very small, right? These are intracellular obligate parasites. These are little machines. Remember, they're not living. They're little machines that live inside of our cell. So obviously they have to be smaller than a cell. Bacteria, a little bit bigger than viruses, right? And then we get up into the different organelles, the little parts, the little pieces of our cells, like a mitochondria, nuclei, are about the same size as most bacteria. And then plant and animal cells are bigger than that. So one of the questions on your study questions here is, which is bigger, a prokaryotic cell or a eukaryotic cell? And we see clearly here, well, first of all, I guess we have to define the two. Prokaryote, pro meaning before, again, the root word pro means before, karyote means chromosome. So these are cells that existed before we had true chromosomes. These are very simple, old, single-celled organisms that are the bacteria or the archaeans. Okay, so prokaryotes here would be on the scale of 100 nanometers to maybe 5 micrometers, versus the eukaryotic or modern chromosome cells. These are cells that have a nucleus that contains chromosomes, and those would be things like plants, animals, fungi, and different type of protists. They're at the scale of up to 100 micrometers. So certainly prokaryotic or old. I just think, again, you need something to get you started. Think of pre or prokaryotic. They're before, they're old, and they're much smaller, single-celled organisms. So the cells themselves are much smaller than eukaryotic cells. Which is simpler, and which is larger, right? We just talked about larger. Simpler, here is a typical bacteria, right? Rod-shaped bacteria, maybe bacillus. And you can see these are relatively simple cells. We're going to look at animal and plant cells later. Many more types of structures inside of those eukaryotic cells. Prokaryotic cells, they have a cell membrane, right? And then outside of that cell membrane is a cell wall. Inside of the cell membrane, there are some things, of course, which are needed for metabolism. There are things that are needed to respond and maintain homeostasis. Those would be things like ribosomes. All living cells have ribosomes, and these are little machines that produce proteins. Prokaryotic cells do have some type of nucleic acid, right? A DNA molecule. So single circular chromosome. Chromosome and DNA are the same thing. Chromosomes are made up of DNA and proteins, right? And there are some enzymes. There's the stuff called cytoplasm that is enclosed inside of the plasma membrane or cell membrane. But and then you have some external type features. These are flagella, like a whip-like structure that propels the bacteria through fluid environments. And there are little pili which maybe help the bacteria stick to surfaces. But again, very simple type of cell, prokaryotic cell. Okay, no nucleus containing true chromosomes, which is older. And there's no doubt about this. These are things called stromatolites. They're these large structures they found off the coast of Australia. And if you look at these microscopically, you're going to see these are fossil mats of ancient archaeans and bacteria. So ancient prokaryotic cells found in huge numbers and have been there for billions of years. Right? The Earth is 4 billion years old, and I think that the first bacteria evolved like 3 billion years ago. So very, very ancient. Eukaryotic cells came much, much later in evolutionary development. Okay. So how do we know that they're older? Again, we can see these fossils of them. Let's see. Here's a side-by-side comparison of pro and eukaryotic cells. We see that on the prokaryotic side, single circular chromosome, eukaryotic cells have one or more linear chromosomes. Again, we have 23 different chromosomes as opposed to one circular chromosome. Age 3.5 billion years ago for pro, 2.1 billion years ago, which is still a long time ago, 2.1 billion years ago, but it certainly more than a billion years younger, the eukaryotic cells. Now, there are some things shared in common by all cell types. So every living thing that we have discovered in our biome on Earth is made of cells, and they're made of either prokaryotic cells or eukaryotic cells. Okay, but all cells have three things in common. One, they all have a cell membrane. Okay, and again, that's sort of what makes it a cell, is that there's a membrane that allows some things in, keeps other things out. It isolates the cell and the environment inside of that membrane from the outside environment, allows them to maintain homeostasis, right? So cell membrane, and then inside of the cell membrane is the cytoplasm that contains ribosomes, right? Those are the machinery that make proteins, and then some nucleic acid, DNA. Okay, so DNA, proteins, cell membranes, those are fundamental to life. All living things express the information in their genetic code into proteins. So proteins are, we're going to talk about all the things they do, but they are fundamental to life. Okay, be able to label plant and animal cells. Okay, so this is the figure 4.3 from your textbook, and I want you to be able to take a diagram and label that with the different organelles here that you can see. So large amount of overlap. Again, we see those three things here. We can see the plasma membrane in the plant cell. We can see the plasma membrane in the animal cell, the ribosomes, and then inside of the nucleus, we'll see those chromosomes. Okay, but then there are a lot of other things we're going to talk. I'm not going to spend a lot of time going through every single organelle, what it does. Later in the lecture, I'll talk about how some of these organelles work together to accomplish their jobs. But you should be able to at least find those organelles on a diagram. Let's see here. Okay. I think this is the last study question for this lecture, right? And it says to illustrate 3 examples of cell dynamism, whereby different organelles exchange materials with one another. Okay, so dynamism means if we look, I guess the point is, if we look at this diagram back here, it looks like a pretty static object here, right? The cell looks like it just stays like this all the time. It doesn't really change. But in reality, cells are very, very dynamic places. There's a lot of stuff going on. These organelles are not independent of each other, but rather they are very interlinked, and they exchange information, they exchange materials with each other. And so I just wanted to kind of show you a few examples of how this works. Okay. So really one of the most important ways in which cells are dynamic. And again, we've alluded to this. All cells have DNA. They all have ribosomes. Okay. And this diagram is a real cursory explanation of why that is. Okay, these proteins over here are shown as this little beaded chain. Proteins do everything in your body. They're the enzymes that help you digest. They're the enzymes that help you to make different things in your body, that make energy in your body. They are immune cells that help you to gobble up and attack and destroy viruses, right? They are muscles. They are messengers. They're hormones that help your body to communicate. So those proteins, suffice to say, at this point in the game, extremely important. How does your body know which proteins to make and when? Because that is coded in the information in your genetic material. Okay, so really this diagram shows why and how you are very similar to your parents. All right, I didn't grow up with my biological dad. He just not around much, but I have learned later in life that actually I'm very similar to him. It's kind of eerie and strange, and the reason why is because on the left side of the diagram, where did I get my DNA from? It came from my father in, not to get too graphic, but in a sperm. Half of my DNA came from my mother in an egg. The two combined, development happens. Here I am, right? So the DNA contains information which is copied in a process called transcription. The message or the copy of that information is sent in the form of mRNA, messenger RNA, out from the nucleus to the ribosome. The ribosome then takes the information in that RNA and translates it into a sequence of protein building blocks. And then those fold into whichever protein is needed. Okay, so DNA has to be there. The ribosome has to be there in order for the protein to be there. And without proteins, cells and therefore organisms just cease to exist. Okay, so a great example of how things are exchanged over space and time, right? The DNA always stays inside the nucleus. The ribosome always stays outside of the nucleus in the cytoplasm. In between them is a membrane, so we need to be able to get the messenger RNA from the nucleus out to the ribosome, right, in order to make that protein. Okay, so I guess this is just showing the same thing. Step 1, transcription. Rewriting the information of the DNA to RNA, sent out through a little pore in the nuclear membrane, right? What its goal is to find a ribosome, okay? A ribosome actually clicks onto the mRNA molecule and it reads along. Remember, because what is DNA and RNA are just a sequence of A's and T's and C's and G's. This is really mind-blowing. Those letters, those 4 letters, are what make you who you are and make you different from an oak tree, right? Because of the proteins that they code for. Okay, so rewrite a transcript as mRNA. The ribosome hops on there, reads those A's, T's, C's, and G's, and then makes a specific protein based on that information. Okay, and that's what makes the world go around. That's what makes your cells work. That makes your personality, your physiology. That determines, honestly, whether or not, probably in a lot of cases, people are susceptible to COVID virus. It's thought that there's a certain percentage of people that are totally immune to HIV virus because of the DNA that they carry and therefore what antibodies they can make. Pretty amazing. Okay, here's another example of cell dynamism. And what we have here just shows, again, here's the nuclear membrane, nuclear envelope. We saw that in the last diagram. Same color, that big purple membrane. Attached to and actually continuous with the nuclear membrane is another organelle called the ER, or the endoplasmic reticulum. Okay, there's the smooth ER and the rough ER. That rough ER is rough because it has little attached and studded into the membrane are those little protein machines called ribosomes. Okay, and so what happens is the messenger RNA comes out of the nuclear envelope and goes directly to the rough ER. And that, what is going to happen in the rough ER? Well, form equals function. What you see there, the shape of something, right, will tell you what it does. Because it's filled with ribosomes, it is involved with protein production. Which proteins? It depends on what mRNA comes out from the nucleus, right? So it's, now the little blue things in the diagram here, the ribosomes are making the little pink things in the diagram, which are proteins. Now, the proteins that are made in the rough ER are not used in the rough ER. They're going to actually be shipped off in these little bubbles to a third or whatever, I don't know, fourth organelle at this point, called the Golgi apparatus. The Golgi apparatus receives those vesicles, takes the proteins, maybe attaches some carbohydrates, maybe folds them, maybe combines them, makes a new product, which is then shipped off the other side of the Golgi apparatus and can be used to do a whole number of things. It may be burped outside of the cell. Maybe you are just slammed a big jug of milk and your body needs some lactase enzymes to break that lactose sugar down. Your body is going to send a bunch of copies of the instructions for the lactase protein. The rough ER makes a bunch of lactase, sends it to the Golgi apparatus. It's modified, finished up, and then it's burped out into your small intestine. And in your small intestine, if you're lucky and you're not lactose intolerant, those proteins will clip up the sugar called lactose. Okay, so again, the point here is that the cell is very dynamic. The parts work together in order to accomplish what's necessary for the organism. Okay, so here's some more of those proteins. They're blue in this color, in this picture. So and what type of protein? These are enzymes. Enzymes are a type of protein. And so you eat food, you chew it up, you swallow it, your acids in your stomach break down some of the proteins. In your small intestine, you've got a lot of enzymes that get in there and break down the big food particles into small food particles. And here, that small food particle shows us this kind of like gross looking, fleshy colored food particle, which is absorbed through the lining of the intestine, goes into a cell, and then those enzymes are combined in a little bubble with the food particle, and you can see digestion happening here, right? And that is just basically taking big particles and making them smaller with the help of an enzyme. Okay, so but those enzymes need to be at the right place in the right time. All starts in the nucleus, the ribosome, the Golgi apparatus, the ER, right? Here, very similar story, except in the small bubble, well, the big bubble, I guess, here, is not a food particle, but a burnt out mitochondria. So the cells are very efficient. If an old organelle stops working, it is made up of those macromolecules like lipids and proteins and carbs. The body or the cell is going to want to recycle those materials. It's going to take those, break them down, ship them off, and then they're going to be used to build something new. Okay, and so how are they broken down? Again, through the process of these enzymatic chemicals, right, that are found in a lysosome. So the lysosome and the vesicle sort of combine, and the enzyme then has access to the old organelle, breaks that down into building blocks, and those building blocks are then used to make a new mitochondria, probably. Right? And really, this idea of cells sharing things is incredibly profound. It happens within a cell. And a great example here is of plants. I mean, plants are really just amazing. And we're going to learn about photosynthesis and cell respiration in some amount of detail. But at this point, just trying to understand, again, this interplay of cell parts. Chloroplast and mitochondria are two different organelles found in cells. We have mitochondria, we don't have chloroplast. It'd be cool if we did, because then we could just photosynthesize. But plants have both. So plants, what they can accomplish is they put their leaf out in the sun, right? Sunlight comes down. It is absorbed inside of the leaf by a pigment called chlorophyll. Uses that energy to drive a series of metabolic reactions that take carbon dioxide, the thing that I'm breathing out right now, and it takes the plant cell, takes that carbon dioxide and builds it, combines it, adds energy to it, and makes sugar. Right? Amazing. Then what does the cell do? It takes that sugar over to the mitochondria, and it takes those chemical bonds in the sugar, breaks them, uses them to make ATP, and then that ATP is used to do everything the plant needs. Right? And we do the same. We eat the sugar from a plant, and then our mitochondria take that sugar and break it down into ATP so that we can contract our muscles, pump our blood. We can use it to generate nerve impulses. Anything that requires work in our cells uses ATP, and that ATP ultimately comes from breaking down food energy, and that food energy ultimately comes from a plant cell absorbing light and making those energetic food molecules. The more you learn about this stuff, I think that you'll start to really find it amazing. So that is lecture 1 of the cancer theme. Again, check out those study questions on Canvas. I hope that they will be useful for you. All right. See you soon.